Hi everybody. Today we're going to be talking about carbohydrates. This is in Chapter 8 of the Fundamentals of Biochemistry by Voigt, and Pratt. This chapter goes over monosaccharides, polysaccharides, as well as glycoproteins. However, for the sake of this class, you're only going to be required to go over the material on sections 1 and 2, which is the monosaccharides and polysaccharides. The glycoproteins is a little above and beyond for this course. It's something we could talk about in a more advanced course, but it is important to understand that proteins can become glycosylated, and that can help a lot with cell-to-cell recognition as well as protein recognition and signaling. But like I said, it's sort of more advanced stuff that we don't really need to go over as of this course. So some of our learning objectives you will see here. Again, feel free to pause, print this out, read over them before continuing the lecture, or read over them after the lecture, read over them after you've read the material or as you're reading material, just so that you can use these objectives to guide you. So, carbohydrates are synonymous with saccharides. One unit is a monosaccharide, as seen here with glucose. When you have two monosaccharides bound together in a glycosidic bond, you can have a disaccharide, such as sucrose. And if you get more than one or more than two monosaccharides bound in glycosidic bonds, you have polysaccharides, as seen in starch. Now, carbohydrates function as storages for energy, as well as in structural materials such as cellulose. And with glycoproteins, we see that we can have them for protein cellular recognition, and so on and so forth. So monosaccharides are derived as either aldoses or ketoses, as you can see here, depending on where the carboxylic functional group is. And we can see that we can have a variety of different aldoses, all of them deriving from D-glycerolaldehyde. So they will all have the stereoisomer formation. As we see in nature, almost all of the sugars in nature are of the D-stereoisoform, and our enzymes are actually incapable of breaking down any of the L-formed sugars. A lot of bacteria do contain L-based sugars, and that's why we can sometimes have a problem during bacterial infections, because our enzymes are unable to break down the L sugars that may encase the outer membrane of the bacterial cells. There's a number of different aldoses. You can have aldotetroses if you have 4 carbons, pentoses, as well as hexoses. But the aldoses that you need to be able to recognize is ribose, glucose, mannose, and galactose. These are your most commonly ones that we'll run across throughout metabolism, as well as other biomolecules. Y'all should recognize ribose from RNA, as well as deoxyribose from DNA. Glucose, is the most common sugar or energy source for the cell in animals. Now we also have ketoses that are derived from dihydroxyacetone, and then D-erythrulose, but the ketose that you need to be able to commit to memory is D-fructose. Now, both ketoses and aldoses can undergo cyclization when an alcohol group further down the molecule reacts with its aldehyde group to form, in the instance of a aldose, a hemiacetyl group. As you can see here with glucose, the hydroxyl group on C5 comes in and interacts with C1, the anomeric carbon on glucose, to form its cyclization. Water is removed. I believe, no, water is not removed. We get a hydrogen transfer, as you can see here. And then we get, this is a 6-membered pyrene ring where oxygen is part of this ring. And depending on where the hydroxyl group is, you can either have the alpha or the beta form of the glycopyranose. These can actually isomerize, however, it's slow, so we can get an equilibrium between the alpha and the beta subunits, alpha and beta rings, in an aqueous environment, although the textbook, I do believe, and I agree, that it is sort of a slow equilibrium. Again, ketoses can undergo the same cyclization, where the alcohol interacts with the ketone carbon to form the hemiacetal. Here we have fructose actually folds and forms into a furan, or a 5-membered ring. And again, depending on the hydroxyl orientation, either below or above the ring, you're going to have the alpha or the beta isoform. Now, as you saw, there are 2 common rings, either the 5-membered pyranose ring, and I shouldn't say 5-carbon, I should just say 5-member, because oxygen is a part of the ring. So you can have the 6-member ring or the 5-member ring. Now, you can have higher order, like 7, 8, or 9-member rings existing biologically. However, they're just not as common. The 5 or 6 tend to be the most common. Smaller rings, such as 3 or 4-membered rings, are very uncommon and do not exist because it's sterically unfavorable and crowded. Now, monosaccharides can undergo a number of different modifications, such as the oxidation of the aldose, which converts the aldehyde to a carboxylic group. This renders the monosaccharide unable to form glycosidic bonds because it no longer has that anomeric carbon. This carbon here is no longer able to react with the alcohol in order to form a glycosidic bonds, or we'll see that further, sorry. Also, you can get oxidation of the primary alcohol group of aldoses, which gives uronic acids. Uronic acids are often coupled with the waste from the human body to make it more water soluble. Usually interacting as a salt form. They can be reduced, and these reduced monosaccharides are often seen in artificial sweeteners. Deoxy sugars are commonly seen, especially deoxyribose, where one of the alcohol groups gets removed. Also, amino sugars, you can add an amine group to these sugars, which then allows for covalent attachment to proteins or lipids to form those glycolipids or glycoproteins. So the glycosidic bond, remember I was talking here, if you oxidize the aldose, you'll no longer get a glycosidic bond, because that bond occurs at the anomeric carbon, which in this instance is this carbon. So the definition for you for an anomeric carbon is, I want to make sure I read it correctly for you in the book, but it is a chiral carbon that has 4 different substituents, and one of them, I believe, being hydrogen. Yeah, so when a monosaccharide cyclizes, the carbonyl carbon, called the anomeric carbon, becomes a chiral center with 2 possible configurations, being the alpha or the beta. So in this case, we have an alpha D-glucose because the anomeric carbon is in the alpha orientation. Now, if we have methanol in the presence of acid, we will see that the methanol group displaces the hydrogen. You also produce water on this condensation reaction, and you form a glycosidic bond with the methane. Okay? Now, depending on where the methane is oriented, you can either have the alpha glycoside or a beta glycoside, again, if it's below or above the ring. Now, some common disaccharides, you have lactose, which forms, you have galactose and glucose linked by a beta 1,4 linkage. So that means you had the beta conformation, so it's above the ring for galactose on carbon 1, the anomeric carbon, on galactose bound to the fourth hydroxyl group, or the fourth carbon, taking place of the fourth hydroxyl group. So the carbon 4 on glucose. Okay? The reason it's angled this way is because in the beta orientation, it's going to be bound above the ring. And on glucose, that hydroxyl group was orientated below the ring. So when it takes place of that, it's going to be bound below the ring. So you end up with sort of a staggered conformation. Sucrose is a glucose with an alpha-1,2 fructose. So again, you have alpha, so it's below the ring here, bound from carbon 1 on glucose to carbon 2 on fructose. And again, that was below the ring, so you have a parallel bond, as you can see here. These are the two, lactose and sucrose, that I want you to be able to recognize and know, be able to tell me what their linkages are and how they're bound together. The rest of these are great examples, however, not something you need to commit to memory for this course. Now, polysaccharides, a very common polysaccharide is cellulose. This is a structural material that we find in such as the cytoskeleton, or not cytoskeleton. This is the plant cell walls, right? So this is what makes up those rigid cell walls of plants, is through cellulose. Now, cellulose is formed from beta, so we're in the beta 1,4 glycosidic linkage between two glucose molecules. Now again, we get this cross interaction, so it's not, this one's in the beta conformation, so it's above the ring, where then it comes and interacts with the next glucose below the ring. So what you end up with is this staggered interaction through the linkage, and it creates planar chains, as you can see here. So here's one chain of cellulose. Now, parallel cellulose chains can form sheets between each other, which can then be stabilized through interchange hydrogen bonds, as we can see here, as well as some positive van der Waals interactions. So these highly networked stabilizing interactions allows these fibers to be exceptionally strong, as well as makes them insoluble in water despite the hydrophilicity of all these hydroxyl groups, because these hydroxyl groups, instead of interacting with water, are already interacting with the pleated, stacked cellulose sheets. Now, although vertebrates themselves do not possess the enzyme capable of hydrolyzing the beta 1,4, see the beta 1,4 linkage of the glycosidic bonds, a lot of animals contain microorganisms such as bacteria that can secrete the enzymes known as cellulases in order to digest these fibers. It just takes a long time. That's why cows, it takes them a very long time to digest the cellulose from the grass and the plants that they are eating, and it's why they chew their cud for a very long time. All right. Now, in plants, the main source of energy storage that they synthesize is alpha amylose and amylopectin. So, alpha amylose is formed of D-glucoses with alpha 1,4 linkages, as you can see here. Because of the alpha 1,4 linkage, it's going to form a helical structure, as opposed to the beta 1,4 linkage of cellulose, which forms those pleated parallel strands that can then stack upon each other to form the fibers. Amylopectin is like amylose. It also has the alpha 1,4 linkages, but it also contains alpha 1,6 branches periodically to create branches in the structure. Now, both of these are synthesized in plants as energy reserve, and can be digested by animals using amylase that's found in the saliva as well as the gut, alpha-glucosidase, and debranching enzymes that can come in and help digest at the branch points. And these are specialized enzymes. Let's see if there's anything else. Now, because of these branch points, you can have multiple reducing ends. As you can see here, ends that are free to either add an additional glucose to, or a lot of these times, these are the ends in which enzymes bind to cleave and then interact to release glucose for energy needs when the cell is ready to undergo digesting the glucose in order to drive phosphorylation or oxidative phosphorylation. And we'll talk about that later on in metabolism. Now, glycogen is how animals store glucose in the cells, and it is a lot like amylopectin but it has more branch points. So amylopectin has branching at about every 24 to 30 residues, as you can see here. However, glycogen can have it every 8 to 14 residues. You get that branch point. So as you can see in B, we have a lot more of these non-reducing end branch points. Oops, well, I guess this is where it starts, so this is where it ends. So the question I ask you is, what is the advantage in the animal cell to store glucose as glycogen with these multiple branch points versus amylopectin? So I'm going to let y'all think about that, and then we will answer that in class on Wednesday. So that's basically a summary of the material that will be covered and the things that are important. Again, some of the key words, again, are like monosaccharide, disaccharide, polysaccharide. Make sure you can distinguish between cellulose and the components of starch, the alpha amylose and amylopectin, as well as glycogen. Make sure you can recognize the monosaccharides such as glucose, ribose, and the disaccharides. Okay? If you have any questions, I'll be happy to go over them with you guys in class, and I will see you then.